Blake, thanks so much for the introduction, and it's great to be here. So glad you could join us. And as you heard, Blake gave you a little bit of an introduction of this research study we're working together with CDI. And my background is really cardiovascular medicine and genetics. And so I'll walk you through some of the concepts, how we decided to really move from genetics into using iPS cells, in particular iPS-derived cardiomyocytes for disease modeling. Here's just my conflict of interest, all the sources which support the research in our lab. And so it's actually kind of interesting to see to be at this automation meeting here because at least in the genetics field, automation and increasing the throughput really gave us a tremendous increase in knowledge. And so we can sequence now. The initial sequencing a human genome was millions of dollars, and now we're down to sequencing a genome in maybe $1,000 or a few $1,000s. So that really shows the great potential of automation and really generating genetic information. But when we look at that, the question is, how can we, what do we do with this type of information? And I'm very much looking forward to Francis Collins' talk tomorrow, so I'm very excited about what he will tell, where the direction goes. Because I think what's pretty clear now is that genetics and really genomic medicine is really a reality, and it really doesn't need any more advertising. I think that it really comes into the clinic, and the word precision medicine is actually now familiar to, I would say, about 31.7 million Americans, because that's the number of people who watch the State of the Union address. And it's kind of an interesting concept if you think that, science and really this cutting-edge science makes it into in a political speech of that nature. But it's really now, I think it really raises the awareness, and I think rightfully so, because I think we really need a new approach in developing drugs and really dealing with genetic information and really recognizing how genetic variation affects, or how genetic variation needs to affect the way how we treat patients. And so, at least from the DNA point of view, we can analyze more and more DNA, we can have more and more DNA samples before, and it actually will only get more. And that was kind of like the background where we started, because if you look at this, we generate a lot of information, but really the question is, if we want to move this to precision medicine, then we need to interpret that. And the slightly flipped thing on the right side should be the latest genome-wide association map. These are all the hits we got from genetic studies looking at complex diseases. We basically have the whole genome now covered with some variants in gene really playing a role for complex diseases. And so there's really kind of like lays the foundation of how we need to go forward with this. So we have all these disease variants, but it's really only statistical association, right? So with GWAS, we identified many loci. Sequencing will just give us more information. But this is really only statistical information. It will only tell us on paper, this is a variant which is associated with that. It doesn't tell us anything how this gene actually really contributes to the overall disease process. And that basically, leaves us potentially with many more questions than that we had before. We're just realizing, oh, well, you know what, this is a gene that has a variation in there. This increases the risk, but what do we do with this? How do we use this in precision medicine? How do we, maybe potentially decide on how we treat a patient differently, what drug we give? We really all need to test that. And so this was really kind of like the foundation of what we do. So the question really what we asked ourselves about a few years ago, and this was really kind of the foundation when we started to work then with CDI, is like, how can we really move from the concept of, at that time I called it individualized medicine, now it's precision medicine, but really it's kind of like more broader approach. How can we really move from the concept to really actually practicing this and including this information? And so I like to postulate that it's really now time to move away from, or move on, take the foundations what we have from DNA sequencing and the statistical association studies to really move on and look at how does a gene function, how does DNA variation function in the context of populations, and really look at the human genome, not just on a G, C, and T level and look at variation, but really look at the whole genome actually at work and how this functions. And so that really, if you want to think so, would be the concept of translational precision medicine. And we really start with a patient, and I think iPSC cells and iPSC-derived cells are really, or will be, the tool to really do this. I can't come up with any other approach. I mean, Blake really told you about animal models and all the other tissue types, what we have. I can't think of any other way where we can really comprehensively look at all the genomic variation, what we see in a individual in a patient, and really study this and look at the effect on any particular disease phenotype we're interested in. So we start with an individual, we generate the iPS cell lines, and then really differentiate those cells in our target cells we're interested in and establish a disease model. And I'll talk to you about these different steps, you know, how we, together with CDI, how we, and part of this grant, how we're pursuing this. But these cells also allow you then not only study this particular underlying disease phenotype, it also allows you to look at related phenotypes. You can really study these cells, you can measure different things, you can look at expression, protein, what you heard from Blake. And so I basically all summarize this in related phenotypes. We can do a very comprehensive phenotyping on these tissue cells. We can use it for biomarker development, which we, again, you know, might not do in large populations. We can do drug screening, and we can do in vitro diagnostics in vitro, basically treating these cells, right? So this is all really nice. I mean, these are all these options, but compared to a animal model, what we really can do is we can actually go back to the individual itself, and we can do in parallel kind of the same what we do in a cell type. So we can do comprehensive phenotype in our individuals, in our patients, in the source. We can do a follow-up analysis. We can follow these patients. We can, again, look at the biomarkers and develop these biomarkers. We can treat a patient, we can diagnose a patient, we give a patient drug. But the potential and the really great advantage here is, what I think, compared to all the other models, what we have, we can actually validate both platforms against each other. A biomarker which I develop in an iPS model is really only valuable for phenotypic screening or for anything else if you can see the same biomarker and has the same role also in your patient. And I think this is really the very unique thing that we can actually go back and forth. And this is also, at least I think, the advantage of our cohort, what we have, and I'll tell you a little bit about our study, how we're pursuing this really parallel approach, modeling in iPS, looking at the patient, and really by that focusing on very clinically relevant phenotypes. So the next step here really is that we need to develop a disease model, and we were lucky enough that we already work with CDI. They put quite some effort into this, so we capitalized on this. So we developed the human cells, and for our disease, I'll talk about left ventricular hypertrophy. It's a pre-stage to heart failure, and I'll show you this example. So we needed cardiomyocytes, and it was luckily enough one of the earliest and first cells what CDI really developed. So we're very fortunate in that way. We wanted to recapitulate the disease. We need to induce a disease state. And by that, it will really allow us to understand disease mechanisms, to look at the inter-individual variability, and then really test for treatment. So those are our goals here. So let me talk a little bit about the disease itself, left ventricular hypertrophy. It's an independent cardiovascular risk factor. It's not that well known. Everybody knows that high blood pressure is not really good. Or diabetes is not good. One of the reasons why we're interested in left ventricular hypertrophy is that it's a risk factor for patients to develop heart failure. It's a pre-stage on the path of developing heart failure. The risk factors are high blood pressure, kidney disease, diabetes, age. Obviously, it's more prevalent in older individuals. Males are at a higher risk, and it's also more prevalent in African Americans. Then also there are a lot of a couple of metabolic diseases and valvular disease, but those are basically the secondary forms. It's not that well known because it's really hard. It's very easy for any individual to measure your own blood pressure. You just get a device. LVH, you really can only determine that. Either you do an ultrasound or you have an MRI. Those are the measurements right now. And so this is why I guess it's not that frequently known about this as a risk factor, but it's a very potent risk factor for heart failure. And to show you this is an epidemiological study published about 10 years ago, where in a large cohort they looked at cardiovascular endpoints. And so in the solid line are the individuals with LVH in this large cohort, a few thousand individuals, and the lighter line are individuals without LVH. And for any cardiovascular endpoint you're looking at, this is a major adverse cardiovascular event, cardiovascular death, which is obviously a pretty significant endpoint, as well as all-cause mortality. For all three endpoints, if you have LVH, you have a significantly higher risk compared to individuals without LVH. So this is really kind of like the foundation why we're really interested in this, and why obviously CDI put a lot of effort into this, and some pharmaceutical companies are actually interested in this particular phenotype as a pre-stage to heart failure. So I told you, and Blake told you this, we have a grant, we're funded through NIH to generate at the endpoint 250 iPS cell lines from patients from our cohort. The overall cohort is about 3,000 individuals. And we have African Americans, it's biracial, and Caucasians. The other interesting component of the study is it's actually a family-based study, so it's not cases and controls, and that has, from a genetic point of view and also looking at risk factor, has some unique advantages, and I would be happy to discuss this with you if you're more interested in this. So the recruitment is we had at least 2 siblings who have high blood pressure, and then we recruited the whole family, so we have multi-generational pedigrees, and that really was the source for us to pick about 250 of the most interesting individuals. We have genome-wide association studies, so all these individuals are genotyped. The initial exam was done about 15 years ago. We have follow-up examinations where very detailed epidemiological characterizations. We just got funded for whole exome sequencing, so we have genomic information. It's a very rich data set. We have serum markers, and we can continuously follow up these cohorts. It's a very rich epidemiological data set, which will really then, what I told in this one slide, build the foundation for the clinical characterization of here, then what we can correlate to the patient cohort. So I'm happy to report, so we're in year 4 of this overall project. So we picked 250 individuals. All 250 samples are reprogrammed into iPS cell lines. That is, to my knowledge, at least one of the largest collections of a diverse set of iPS cell lines of patients to date. And so we're very excited about this. We recruited about 290 individuals. All these lines are banked and fully characterized. And obviously, you not only can make cardiomyocytes out of this, you can also make additional types out of this. So we're looking also potentially if NIH is going to fund us for hepatocytes and endothelial cells and so on. But anyway, we're focusing mainly on cardiomyocytes. So Blake showed you this slide here before. This is where we are. So we have now about 120, 130 cardiomyocytes lines made out of these 250. And this is the graph here shows about the quality criteria of the first 90 or so individuals. And you can see that for all individuals, we get over 90% pure cardiomyocytes. And kind of Blake showed this to you very briefly or cursory. Let me mention this, how really important and groundbreaking this really is, because if you talk to people in the field who make cardiomyocytes, generating the number of cardiomyocytes for any individual who comes in at this purity is pretty unheard of, because you have a lot of variability in the yield of cardiomyocytes. You have a lot of production runs where you maybe get about 60 or 70%. There's a lot of variation what you introduce, obviously, by studying different individuals. And so the work which went into this to generate these high purity for all these cell lines, for every individual who basically comes in, is pretty unique. But this really lies the foundation for these studies where we're going to compare different individuals, because if you introduce variation already at the variation from the process how you make cardiomyocytes, that bleeds all the way through your analysis when you look at the effect between different individuals. So you need to minimize the variation you introduce just by making these different cell lines and making these cardiomyocytes. And so I think this is one of the key accomplishments and the key data. When I saw this, I was actually quite excited about this. So this is really a major accomplishment on the side of CDI. So I told you about the development of the hypertrophy assay. So we induce hypertrophy with different inducers. So mainly you saw that Blake showed you endothelin-1 stimulation. We also tested angiotensin-2 or and isoproterenol. Those are all well-established inducers of hypertrophy. These cells really respond to all this and to various degree actually change their expression pattern. So we did immunofluorescence. This is again looking at CFOS and CMYC. These are also early immediate genes which are differentially expressed besides BNP as a marker for hypertrophy. These cells increase in cell size, as you can see here compared to the controls with a different stimulant. If we look at CFOS immunofluorescence, we also see an increase. So these cells actually, using different stimulant, develop hypertrophy. We developed, and for our studies, we developed the assay in a 96 format. I recognize that, here in the throughput 96 format might not even sound that high throughput. But we are really interested in a very comprehensive assay, so we would like to, we are stimulating these cells. We also get protein. We want to have serum. We look at RNA, and we needed enough material. And we're stimulating these cells in different ways. So we stimulate the cells with endothelin, phenylephrine, isoproterenol. We combine this. We have a couple other ways to really characterize these cells. So we, to obtain enough material for RNA sequencing in a downstream analysis, we decided to run these experiments right now in 96. You're not restricted to this, obviously. 384 just gives you less material downstream. So we spent a lot of time on optimizing this, and the assay does not only work in the hands of a company who really devotes their work to this. So we did over extended period of time. We took these cells. The results of our stimulation protocol highly reproducible. So we did that. The other thing we spent quite some time in working on really characterizing the results, what we see. And so one of the questions what I always get is like, well, how does this actually compare to what you see in a patient? And so we are interested particularly in expression analysis. And so what we did is we did just a global expression analysis, and we were lucky enough to have expression data from patients, individuals, from where cardiomyocytes or biopsy data was obtained, and they had expression data on these patients. And we have expression data from patients, normal patients, patients with left ventricular hypertrophy, and patients with left ventricular hypertrophy and heart failure. And so what we did is we basically compared the expression changes, what we see in the iPS-derived cardiomyocytes, and we compared the expression changes, what you see in patients with hypertrophy from a cardiac biopsy. This is really live patients' cardiac material. And so one way to really look at that, let me introduce you to this, is basically, well, you can look at all these different genes which are the same or which are different. But one way to really reduce this and really compare this is called principal component analysis. And you're just basically looking at overlap and similarities. And so here, this is basically principal component 1. This is the separation, the main source of variation. And if you look at this here are the blue and the yellow dots, and those are the cells from iPS-derived cardiomyocytes. And here on the left side is the data from the patient biopsy data. So the largest variation is obviously iPS-derived cells and biopsy data. That totally makes sense, at least to me, because a human is a human, and cells and cardiomyocytes, it's only one cell type. The cardiac cells from the heart are obviously a lot of different cells. There are also fibroblasts in there. There's cardiac matrix in there. So there's a lot more into this. But the second principal component, the separation here from top to the bottom here, basically separates out normal control biopsy and the normal iPS-derived cardiomyocytes to the cells which are hypertrophic. So the second largest source of variation, if we compare iPS-derived cardiomyocytes and biopsy data, is really the presence of hypertrophy. And we have a large list of genes which are, again, the same way regulated, upregulated or downregulated, both in these cell types, in human cells and iPS-derived cells. And that gives you a good reference set and really shows that these cells do in fact recapitulate on an expression level the disease phenotype what we see actually in patients. So we're very close to this. We also did this experiment actually with rat cells. It doesn't really look that great, but there are some challenges in generating this. But what I really want to show here is that these cells actually do recapitulate on expression level the phenotypes we see in patients. So we reprogrammed that. So we did a first round of phenotyping. Now I told you that we're looking at a large number of individuals. So the cardiomyocytes are slowly getting in the lab, and we're studying them. And so the first question, obviously, what we have is, if you assume that these cardiomyocytes and these iPS-derived cells recapitulate the phenotype, do we see any phenotypic variation between different individuals? Right? I mean, they could be all the same. Wouldn't even see. I mean, we have all these different individuals. Some have risk for LVH and extreme LVH. Some have not so much LVH. They're actually normal. Do we actually see phenotypic variation? And in fact, we actually do. So here are the dose response curves for all the list of these cell lines here. These are, this is the I cell from CDI, and these are all the patient-specific, cohort-specific individuals. And you can see here quite the variation in fold change. This is BNP expression over the range of the stimulation with endothelin endothelin-1. So you can see here that, we have some cells which are quite sensitive and increase their BMP expression quite a bit. We also have a few cell lines which are not very impressed, with endothelin-1 at all. We also looked at the stimulation I showed you. We stimulate not only with endothelin-1. These are results with isoproterenol and phenylephrine stimulation. You can see here that we also see quite a phenotypic variation between different individuals, as well as if we stimulate the cells with endothelin-1 or norepinephrine. So we do see phenotypic variation in these cell lines, and we obviously expect that because we see a phenotypic variation between these individuals. So there's quite some substantial phenotypic variation. Now, one of the questions what I always get is like, well, does this now really recapitulate what you see actually in your individual? And obviously, I would be, I'm much more confident to answer this question when we study all 250 cell lines. That will still take a little while. But we did a very preliminary analysis where we basically looked at all these cell lines which were studied so far. And when we do a regression analysis, we basically compare right now the EC50 to our endothelin-1 stimulation with a number of different phenotypes what we see. And I told you in the beginning we have the first exam done about 15 years ago on these individuals. We did actually another exam just at a time point when we recruited these individuals, again about 2 years ago. So we have about a 13-year window, and we know their LV mass 15 years ago, and we now know their LV mass about 2 years ago. And we can look at the change over time. And when we look at the change over time, this change over time, I would say at least kind of like suggestively correlates with the EC50 in these cells. And now, do I really strongly believe that these results are, this is really very solid? I think we need more cell lines, we need more individuals, but at least it gives you a hint of what type of analysis we can do. We can basically correlate phenotypes, what we see in the iPS-derived cardiomyocytes, with the phenotypes and change over time what we see in the individuals here. So we're going to increase, obviously, the sample size, and we're going to do this analysis and continue to do this analysis. But it fits overall in this concept what we observe with a couple other studies, that there are some individuals who are more susceptible to environmental risk factors and develop LVH, and there are some individuals who are less susceptible, and there might be more, there are some iPS-derived cardiomyocytes who you can tickle a little bit more and they develop hypertrophy much more, and there are some iPS-derived cardiomyocytes and you can tickle them quite a bit and they actually are pretty unimpressed. So I think this really fits in the overall concept. Now, I told you that we're interested in developing additional biomarkers, and so one of the biomarkers we were really looking at, and that's really related to a lot of the knowledge, what we think and what we learned over the last few years about the role of small RNAs, in particular microRNAs in cardiovascular disease. And that's just a brief overview of a review from a few years ago of which Eric Olsen published, and the role of microRNAs, how they work. They are small RNAs. They form typical structures here, and they basically bind to RNA and basically downregulate the expression of RNAs and play a large role in the development and contribute to cardiovascular disease, in particular LVH. So there are a number of different methodologies out there how you can measure microRNAs. There are some arrays out there, and you have these preconceived and established RNAs. MicroRNAs, we decided to actually do some sequencing because we think that there are a lot of variants out there, a lot of microRNAs out there which are really not annotated yet. And so we did microRNA sequencing, and we published this just about a year ago. So if you want to have look at the details, we sequenced the microRNAs. We took these small little sequence reads. There are programs out there which allow you to predict the base structure, allow you to take the reads, predict if they are microRNA or not. And so we ended up with a number of these predicted microRNAs which are also differentially expressed, and we compared expression normal cardiomyocytes with the hypertrophic cardiomyocytes. So we found quite a significant number of differentially expressed genes. We started here with a certain list. Some of them were known, some of them are predicted, and we were particularly interested in the predicted ones, the ones which are not known as a microRNA, not in these databases established as that. So the next question was, are these really real? Because obviously if a computer program tells you this is something, you're not really, very you're not really convinced that this is really actually true. And so one of the ways then, again, which allows you, the iPS-derived cardiomyocyte platform really allows you to take the next step and really functionally validate that and really move from gene sequence to actual functional validation. And so these are just some experiments. I'll walk you through those here, where we had 2 predicted microRNAs. They looked by all means very much like a microRNA from their structure. They had certain targets, and so we designed experiments to knock down these microRNAs in the cardiomyocytes. Here it is, just these are the levels, the microRNA levels when we designed siRNA to knock those predicted microRNAs down here. And these are then where we looked at the same time we looked at the expression of BNP in those cells. When we knocked these particular microRNAs down, which have been differentially expressed, and those are duplicate experiments. Here, the column on the right is always the expression of BNP with endothelin-1 stimulation, just the control siRNA. And for both microRNAs, these predicted ones, I'll show you those 2. If we knock down these microRNAs, the cells do not develop hypertrophy. They do not increase their expression as much as the control. So that gives us a pretty strong evidence that these microRNAs actually are, in fact, playing a role in the pathways which lead to hypertrophy and the increase in BNP expression as we knock these microRNAs down and prevent the increase the development of hypertrophy. The other thing, we looked obviously at overall gene expression in these knockdown experiments and did pathway analysis, just a very cursory show you a number of genes which have been differentially expressed when we knock down these microRNAs, play a role in dilated cardiomyopathy, in vascular smooth muscle contraction, and calcium handling. So they actually hit certain pathways which are linked to this development. And the final slide here on microRNAs. So one of the questions obviously is there's a lot of interest in microRNAs as diagnostic markers, particularly in the field of cancer, and I think in the cardiovascular field we're also recognizing this. And so we actually looked at a number of different cell lines to see, again, different from different individuals from our cohort, to look at the expression of these particular microRNAs. And we also included a known microRNA. So MIR1 is a known microRNA playing a role as established microRNA in development of hypertrophy. And we see here between the different cell lines, and this here again is the I cell, we see quite some variation again in the level of the expression of MIR1 in different cell lines. Again, really directing to the idea that these cells actually do recapitulate phenotypic variation. We also see this for our 2 predicted novel microRNAs. So we have quite some variation here in these cells. As we're interested in biomarkers, the way how it's conceptualized right now is that microRNAs get actually excreted from a cell into the surrounding media. So there's a lot of interest in looking at microRNA-free microRNAs in serum samples, again in cancer. So we're moving in the same direction. The first question we asked is, well, we find these microRNAs differentially expressed in the cardiomyocyte. Are they also getting excreted from the cardiomyocyte? Now, if we have it in cell culture, it would be in the serum media. And in fact, they actually do. Here is the data of these microRNAs excreted as part of exosomes in the cell culture media, so we see some variation there as well. And we just extended in our study, we also looked in our cohort of patients if these novel microRNAs are in fact present in the serum of our patients, and they actually are as well, with some variation here too. So we move from differentially expressed microRNAs to microRNAs excreted from the iPS-derived cardiomyocyte in the cell culture media to these microRNAs excreted and present also in the serum of patients, which were the source of these iPS-derived cells. So that gives you a rough idea how we would like to, how we're conceptualizing moving from the tissue again back to the individual and comparing and looking at these biomarkers. So very briefly, the other question what we got pretty frequently is, so when we're looking at different individuals, do we actually see this phenotypic variation in how we handle these cells and how we stimulate these cells? Do these cells retain their individuality? And so this, again, just really shows you an expression analysis, what we did in a principal component analysis, where we took right now 4 different cell lines, and we basically looked at the expression when these cells are unstimulated. So it's basically like baseline when they're just in the dish, and when we stimulate them. And now here again is the result for the endothelin 1 stimulation. And on the right side, you see here the principal component analysis. And when we did this hierarchical clustering, you can basically see that these cells, or these different experiments cluster in about these 8 experiments, expression analysis cluster in 4 different groups. And the closest one are always the individuals. This would be unstimulated. This is the endothelin 1 stimulated. This is one individual. This is one individual. There's an individual in here. So both by the phenotypic variation of what we see, they cluster obviously whether we induce them or not, cluster closely together, and we really capture their unique individualness. If you want to think of it this way. When we do this in the principal component analysis, you see this all as well. So oops, sorry, that was the wrong button. If we look at the different individuals, you see every individual basically clusters for itself. We pick those individuals to represent certain parts and certain features of hypertrophy. So you see that they're clustered in their own little space, but they're still similar to each other whether we stimulate them or not. Now this really kind of like shows you that they're all these 4 individuals are different. We picked them to potentially really have a phenotypic spectrum from our first few cell lines. What we have, if you think down the road, if we have 250 individuals, what I would postulate this dendrogram or this histogram is going to look like, that we have groups of individuals who are going to have, who basically fall kind of like in similar clades, like we have these 2 here and these 2 here. So that's a way, at least how we conceptualize this at the moment. This is a way how we, on a molecular level, subgroup phenotypically patients based on the expression level. So this is just one way how we can look at this, and also then take this type of data and define then certain disease entities. So let me go to the next step, and this is a collaboration with a group in Bang at Sanford Burnham. We are really obviously also interested in using these cells, and not just one cell line, but multiple cell lines or maybe subgroups of these cell lines based on the phenotypic clustering, what I showed you in the previous slide, to use this for phenotypic screening. And so the first question was, can we actually use these cells for phenotypic screening? I think at this meeting, I'm the one who's learning more about screening here, so you probably all know more about screening and developing high-throughput assays using these cells. So as we're diving into this, we're again looking at BNP expression with high-content imaging. This is here the control cell line. This is basically we stimulate the cells, and we're looking at cell size as well. And we again use endothelin 1, but we extend this also to a couple other stimulants. So we basically see here that the BNP expression using high-content screening obviously increases as well as cell size. And if we use a drug which is, which blocks endothelin 1 in a control, that would be macitentan at this time, bosentan at this time. It's an endothelin 1 antagonist. You can basically see that we can, bring the expression down to the control levels here. So overall, the C factor for that screening was about 0.5, 0.6. So we were pretty content and happy with this. And so we just finished, and this is right out, from our analysis, we just finished a first screen with a small library. The Presswick library is about 1,200 FDA-approved drugs. We also did a screening for kinase inhibitors. Obviously, kinase inhibitors play a key role or implicated in the development of hypertrophy. And just want to, as we're sifting through this data, want to point you to a couple things. Obviously, we have the vehicle controls here. This is the the screen was done in 384 well plates. We did it this is really just a primary screen at a concentration of 5 micromolar. We included macitentan, that's another endothelin 1 antagonist, as a control. And some of the hits will basically look at the reduction of BNP expression under stimulation. Would be bosentan, the drug I showed you before, or ambrisentan, that's basically a similar one. We did this. We did the screen also in duplicates, so these are the duplicates, the dark one and the lighter one. And so we both have these compounds showing up as potential interesting hits, but you also see that we have a few other ones which show up here. And we hit a number of drugs which we obviously expect because they're used to treat patients with hypertrophy, but we also ended up with quite a few hits, which, at least from my clinical perspective, I would not prescribe a patients with hypertrophy. So we're actually quite excited about this. That obviously needs still some follow-up, with different concentration dose response, and we're also going to look at this at in different cell lines to really have a robust set. But we're very excited about these initial results, and obviously we would also be interested in using these cell lines then for the novel drug development. But at least from a clinical point of view, these FDA approved drugs are a very elegant way to develop new drugs. So the other thing, what I want to show you here real quick is, so this was one cell line. We also analyzed a second cell line, which is a patient cell line. And as Blake told you, when we're looking at the patient with a monogenic form of hypertrophic cardiomyopathy, they have similar expression patterns. This is another patient where we compared a patient from our cohort. This is not a monogenic form. This is really a polygenic form. This is what we're recruiting. And we can actually look at, again, baseline expression of BNP and compare this to a control cell line. And this is what we did here. In gray, the patient, in dark, the control. And you can see here, even without stimulation, the iPS-derived cardiomyocyte from the patient with LVAs has increased baseline expression and of BNP. So kind of like the concept, these cardiomyocytes from patients who are susceptible are maybe more ticklish, or you can induce this, already have a higher baseline expression or platform expression compared to the controls. And when we treat these cells either with macitentan or bosentan, which is the more widely used endothelin-1 antagonist, we can basically normalize the expression of BNP to almost control levels. So at least in the dish, we can treat this patient with an endothelin-1 antagonist and normalize the BNP expression. Now I can tell you that endothelin-1 antagonists currently are not the drug of choice to treat a patient with hypertrophy. They were initially developed to treat patients with hypertension. They're now used only in patients with primary or pulmonary hypertension, so high blood pressure in the lungs. They're not really used for this. I can also tell you that this patient, which was the source of this material, is not treated with this drug. Now, would I go that far to tell basically that, if you're this patient here and you're in the audience, you should go to your doctor and tell him that you need an endothelin-1 antagonist? I'm not really sure yet, but this is certainly, I think, one of the concepts we're really trying to validate and test. And what I showed you in this one slide, how we can treat these cells and again then go back to the patient and really look at the correlation between those two things. So with that, let me just summarize here my presentation. I hope I could convince you that these iPS-derived cardiomyocytes really do resemble the phenotype in the disease state, what we see in patients. And human disease phenotypes, that we can use these cells for the functional analysis, which can lead to the identification of novel disease markers like the microRNAs, and we can actually take this then again and go back to the patient individuals to validate these markers, not just develop this in a cell culture, but we really have the potential, like going back, really validating this and looking at the impact of this, and if this is really a true valid biomarker. We see that these iPS-derived cardiomyocytes show, in fact, phenotypic variation, which is really relevant to clinical phenotypes, and I think that's still in the whole field quite up for debate. So I think not just in cardiomyocytes, also the program I'm participating where, a part of this funding mechanism, we're really looking at that and making strides not only in cardiomyocytes, but in also tissue and different tissue types. We're really excited because I think it really provides a very unprecedented platform for functional genome analysis. And I didn't really dive into sequencing and some of our gene editing efforts. We have one cell line, which we're studying right now, which has been gene edited for a highly likely candidate, which we identify through whole exome sequencing, and really can incorporate this into genomic and phenotypic variation analysis. So we're looking forward to those results. And really the evaluation and the validation of, in the comparison to the human disease phenotypes, and having both human and iPS cell line together, I think is really, in our opinion, emerges as really the most interesting and exciting aspect of iPS disease modeling, particularly if you're interested in phenotypic screens. So with that, let me just mention all the people who really work on this. In my lab, basically Andrew and Amy are doing the IPS experiment together with Aaron. Broeckel is our bioinformatics person who worked a lot on the sequence analysis for microRNAs. My collaborator is a Hypertrophy. This is an ongoing study funded by NHLBI. It's one of the NHLBI cardiovascular cohorts. Particularly John Arnett and Charles, who is a statistical person on this. Definitely would like to thank all the collaborators on CDI who are co-investigators on our NIH grant. Emil, Blake, Chris, Steve, and everybody who works in the IPS team. It's a really large effort to derive 250 iPS cell lines and make cardiomyocytes out of this, and I think it's certainly a new adventure for CDI as well. And last but not least, I also want to mention our program person at NHLBI, Kishel Jaquish. She really shepherds not just our program, but 8 other programs through this. It's one of the most ambitious monetary commitments of NHLBI to IPS disease modeling, and so she is certainly spearheading this, and we're very grateful to have her really direct that. And sometimes people at NIH and NHLBI don't really get all the recognition for what they do, so I definitely would like to mention her. And so I hope I could enlighten you a little bit, and I will be more than happy, I guess, together with Blake, answer questions. Thank you.